Hello, this is Mr. Hagge, and today I'm going to show you the setup and procedures for measuring the cell respiration rate, aerobic cell respiration rate in germinating peas. The setup consists of two respirometers, and these are respirometers that I have designed and built, consisting of plastic tubing, which has been molded, bent into that form a U-shaped respirometer. Transparency film with graph paper copied onto it. And the graph paper is in 5mm increments. We have a plastic straight tube with a rubber hose connected that is right now open to atmosphere. And so we have two of those manometers. The one manometer will go on the experimental tube, which will have the peas. The second manometer will go on our negative control, which will be non-germinating peas, which will be plastic beads, simulating non-germinating peas. The test tubes we're using are Chemex, Pyrex Chemex tubes, 25 by 150 millimeters. We also will need some absorbent cotton. We're using two Hoffman tube clamps, using 15% potassium hydroxide, and the potassium hydroxide is used to absorb any carbon dioxide that will be produced during aerobic cell respiration by the peas. Our manometer fluid is glycerol and water with some green food coloring added. The transparency pen for marking your fluid levels in the respirometer, and then a little spatula, scoopula for pushing our cotton. So to set this up, first thing you'll need to do is add non-absorbent cotton to the bottom of each tube. Again, we do the same for both setups. Then we'll need to add 15% potassium hydroxide to the cotton. You need to really soak down the cotton. I've already kind of pre-soaked the cotton there, so we'll just add a little bit more here. But the cotton needs to be soaked so that you see that it's obviously moist and wet throughout the cotton. Now the second cotton we're going to add and push down into the tube so that it's just hovering above the cotton with the potassium hydroxide. You don't want it to touch that because you don't want it to absorb any of the potassium hydroxide. That cotton there is to protect the seeds. And also when you're adding the potassium hydroxide, make sure you don't dribble it on the side of the tube because the potassium hydroxide is pretty caustic, 15%. And the same thing was done to the control tube as well. Okay, so next we're going to add fluid to the manometer or respirometer, and I've already added it to the control tube, and I'll add it to the experimental tube. And I used a plastic pipette because it fits the inside diameter of the tube better. And so we need somewhere between a half and 1 milliliter of fluid. Let's put that down in there. Oops. If it does that, we want to make sure our fluid, we don't have any air bubbles or fluid up in the tube. So what I'm going to do is just push air in, up and down here to push the fluid up and down until we get all the air bubbles out. So, okay, good. And then again, we need enough fluid to make sure it's come up around the U and is up on the grids of the graph paper there. Close up the lids here. The next thing we're going to do is add our germinating peas to the test tube. And I'm going to add enough germinating peas so that it doesn't completely fill the tube. You need to leave some airspace at the centimeter or so, a couple centimeters, so that the tube does not come in contact, or the stopper does not come in contact with the germinating peas. Okay, so we're going to add Again, make sure these peas are very well germinated. You see obvious signs that they have germinated in their little new roots here. So it looks really good. So again, the more you pack in there, the better your results. But you definitely want to leave that airspace at the top there. Thermometer on here. Need to make sure that has a tight seal. Okay. Now to the second tube, our control tube, we'll add the same volume of our plastic peas, our non-germinating peas. And I like these because they are about the same size as the germinating peas. And it's going to do a good job as our negative control. That same volume of peas in both tubes. And that's pretty close there. Okay. Add our manometer. Go here. The next thing I'm going to do is measure our starting level in each manometer, respirometer, and this one's already marked here, so we'll get this marked. Now remember that it's going to be using oxygen as it does cell respiration, so the fluid is going to be drawn to the tube, because what we're going to do is we're going to close this off and create a sealed chamber. And so as the oxygen is being used, it will flow toward the peas, the manometer fluid. Now again, make sure you get down here at eye level so you get a good accurate marking of the level. Okay, now I will let it set at atmosphere unclamped, give it like 3 minutes to set and equilibrate. Then after the 3-minute time period, then we're going to clamp off both tubes and start a timer. And we're going to do a 30-minute run. At 15 minutes, we'll mark the level that the fluid has risen in the respirometer. And then again at 30 minutes when time is up. So we'll clamp these off, so we'll again, though we want to give it 3 minutes to equilibrate before we clamp them off. Now, at the end of the 30-minute run, what we'll do is measure the amount of oxygen used by measuring the level that the fluid has risen. And so our tube is a cylinder. So we can measure the volume of the cylinder to determine the amount of oxygen gas being used. And so the volume that the fluid rises will equal the amount of oxygen used. And so we'll use our volume of a cylinder, which is the area of the tube, which is pi r squared, times the distance the liquid has moved in the tube after 30 minutes. Let's zoom in here and get a good look here at the setup. If you are interested in this experiment, please don't hesitate to email me. You can email me at tshagge@yahoo.com. That's tshagge@yahoo.com. And I would be glad to send you my experimental procedure, and then also talk to you about ways that you can get my respirometer. Again, this respirometer and my manometer can be used in photosynthesis experiments, which I have a YouTube video right now describing the photosynthesis experiment as well. Zoom in here. As you can see, we already have some movement. Some nice movement already in our germinating peas. And